Good morning. It's May 16th, and this is your daily brief in science. Here's everything you need to know. Researchers at Stanford Medicine have made a significant breakthrough in feline genetics by identifying a genetic mutation responsible for the distinctive orange coloration found in domestic cats. This trait is sex-linked, meaning it is more prevalent in males due to their single X chromosome. The mutation involves a small deletion in a non-coding region of the X chromosome, which activates the nearby RGAP36 gene and alters pigment production. Analysis of skin tissue from calico cats revealed that the RGAP36 gene exhibits increased activity in the orange patches compared to black or white patches. This suggests that the deleted section typically suppresses the gene's activity. Professor Hiroyuki Sasaki, the lead author of the study, expressed excitement about uncovering the long-sought orange gene after years of research, emphasizing its significance in understanding feline genetics. The findings contribute to a broader understanding of gene interactions and their implications for development, which could enhance insights into gene regulation in mammals. Geneticist Chris Kalin noted that this trait's sex-linked nature affects its prevalence, particularly in male cats. The research confirms that the orange coloration is a result of a mutation affecting gene activity rather than a specific orange gene. This investigation into the genetic basis of orange coloration has broader implications for understanding pigment type switching across various species. While the study raises questions about potential links between coat color and behavior, initial findings did not show significant differences in gene expression outside of pigment cells. Additionally, the research may provide insights into whether orange cats are more susceptible to certain health conditions, given the ARH-GAP36 gene's association with other body areas, including the brain and hormonal glands. Supported by the National Institutes of Health and contributions from various universities, the study reflects a growing enthusiasm for feline genetics, having raised over 10 million yen from cat lovers. Roche has recently unveiled 10-year results from the Phase 3 APHINITY study, which show a 17% reduction in the risk of death for patients with HER2-positive early-stage breast cancer treated with a Perjeta-based regimen compared to a control group. These findings were presented as a late-breaking abstract at the 2025 ESMO Breast Cancer Congress, highlighting the significance of this long-term data. The primary endpoint of the APHINITY trial focused on invasive disease-free survival, with secondary endpoints assessing overall survival, disease-free survival, and safety. Genentech's chief medical officer Levi Garraway emphasized the sustained benefits of the Perjeta-based regimen, reinforcing its status as a standard of care treatment in curative settings. The safety profile of the Perjeta-based regimen remained consistent with previous studies, showing no new safety concerns and a low rate of primary cardiac events. Roche continues to project significant revenue from both Herceptin and Perjeta, with expected sales of $4.3 billion for Perjeta and $1.6 billion for Herceptin in 2024. Perjeta, which received FDA approval in 2012, is now utilized in over 120 regions for treating both early-stage and metastatic HER2-positive breast cancer. In the APHINITY study, participants were randomly assigned to receive either trastuzumab deruxtecan monotherapy, trastuzumab deruxtecan followed by THP, or standard care. Pathologic complete response indicating no invasive cancer cells remain after pre-surgery treatment is a critical goal in breast cancer therapy. AstraZeneca's Susan Galbraith noted the potential of ENHERTU to challenge current treatment standards and enable earlier intervention for potential cures. Given that HER2-positive breast cancer presents a challenging subtype, with a third of early-stage cases at high risk for recurrence, innovative treatment solutions like the Perjeta-based regimen are essential. Recent research published in the Cryosphere has unveiled alarming acceleration of glaciers in West Antarctica, particularly in the Pope, Smith, and Kohler regions. This acceleration raises significant concerns about global sea level rise, as the Antarctic ice sheet, once thought to be stable, is now recognized as changing rapidly and unpredictably. One notable finding involves the Kohler East Glacier, which has been observed stealing ice from the slower Kohler West Glacier, a phenomenon referred to as ice piracy. This process has substantial implications for ice mass dynamics and is crucial for understanding contemporary changes in ice sheets. Changes in ice flow direction have been shown to significantly impact mass flux into critical ice shelves, including the rapidly changing Dotson and Crosson ice shelves. By 2022, 6 ice streams in the region were moving at speeds exceeding 700 meters per year, indicating potential instability within the ice sheet. From 2005 to 2022, significant acceleration was recorded in 7 ice streams, with one glacier's speed increasing by 87% at its ocean meeting point. The study highlights the influence of changing ocean conditions, air temperature, and snowfall on glacier dynamics and the redirection of ice flow. This unprecedented discovery challenges previous beliefs that significant glacial changes took thousands of years, as these shifts have now occurred in less than 18 years. Researchers utilized high-resolution satellite data to document these rapid changes, revealing that processes once thought to take centuries can now transpire in under 2 decades. Projected future sea level rise could affect over 410 million people by 2100, with global sea levels having already risen over 10 centimeters in the last decade. The use of satellite technology has revolutionized climate research, enabling scientists to monitor and predict changes in ice sheets with unprecedented accuracy and detail. A significant breakthrough in breast cancer research has emerged from South Africa, where a groundbreaking study has identified genetic variants associated with breast cancer, specifically in Black South African women. This marks the first genome-wide association study, or GWAS, of breast cancer conducted on the African continent, addressing a critical gap in the understanding of the disease. Co-lead author Dr. Jean-Tristan Brandenburg pointed out that the scarcity of data on African populations complicates accurate disease risk prediction. The research team believes that identifying these genetic variations could pave the way for more targeted cancer treatments, allowing for therapies that spare healthy tissue, as explained by Prof. Chris Matthew, the lead investigator at the Sidney Brenner Institute for Molecular Bioscience. The study's findings may lead to the development of targeted drugs aimed at the newly identified genes, potentially enhancing treatment precision by focusing on harmful cancer cells while protecting healthy tissue. Prof. Matthew also highlighted that understanding the role of genes like USP22 could help identify patients who might require more intensive treatment and closer follow-up. If these genetic markers are confirmed, they could serve as biomarkers to predict aggressive forms of cancer, guiding treatment and monitoring strategies for patients. Dr. Hyatt emphasized that the findings indicate the potential for discovering new risk factors, which could ultimately benefit more individuals in understanding breast cancer. The Institute also recommends regular mammograms, self-checks, and maintaining a healthy lifestyle for early detection and treatment of breast cancer. Notably, RAB27A is part of the RAS oncogene family, while USP22 is linked to poor prognosis in breast cancer, underscoring their potential role in the disease's genetic basis. A recent study sheds light on the cognitive processes behind those enlightening aha moments we all experience. Published in the journal Nature Communications on May 9, 2025, this research highlights how such moments can significantly enhance memory retention. Researchers from Duke University, along with German institutions, discovered that the hippocampus, a critical region for learning and memory, exhibits increased activity during these insightful moments. This boost in hippocampal activity correlates with stronger memory retention, suggesting a profound connection between insight and memory. The study involved participants solving visual brain teasers while their brain activity was monitored using functional magnetic resonance imaging, or fMRI. Findings revealed that individuals who experienced sudden flashes of insight were nearly twice as likely to remember the solutions compared to those who relied on methodical reasoning. Additionally, changes in neuronal activation patterns were noted in the ventral occipito-temporal cortex, indicating a reorganization of information processing during these insights. Researchers linked stronger insights to improved connectivity and communication efficiency among various brain regions, which enhances overall cognitive processing. The implications of this study suggest that creating environments that foster insight could lead to better long-term memory and learning outcomes, particularly in educational settings. Looking ahead, future research aims to explore the brief moments between the onset of insight and its realization to deepen our understanding of the creative process. First author Maxie Becker emphasized the significance of these moments, stating that experiencing an aha moment nearly doubles the likelihood of remembering a solution. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.